To my mind, the most important revolution in the last 100 years really has been the sequencing of the human genome, because that provided the catalogue of all the proteins that exist in a cell. It identified who we really are. It didn't tell us how we work, but it told us what we're made of.
A key assumption that underpins our understanding of cells is that proteins with shared functions end up in membrane-bound compartments. This has been challenged by ever-increasing evidence for compartments that are not membrane-bound and contain proteins of shared functions. These non-membrane-bound compartments, sometimes called condensates, are able to compartmentalize and concentrate components that have shared functions.
One of the reasons we've been so interested in condensate biology is that condensates explain how all these individual molecules work together as collectives.
I think this discovery will change our understanding of the science of a cell, and therefore will change how we view the treatment of disease.
What's very exciting is that many aspects of disease which we didn't understand before are now suddenly becoming clear. We've seen that quite often disease arises when these condensates become aberrant, and this provides a whole new way to think about intervening and preventing these disease-like states.
Another set of diseases are these neurodegenerative diseases such as Alzheimer's and Parkinson's. One of the common features is protein aggregates. So tau aggregates, amyloid fibrils, amyloid plaques, or Lewy bodies. All these are protein aggregates. And the interesting issue is, are these condensates? Can we look from the angle of a condensate in a different way at these diseases? And again, we don't know the answer. But we do have the tools to look at this, and that is very exciting.
The most important elements of gene control for each cell are occurring in a three-dimensional condensate in which DNA is wrapped up in 3 dimensions. But what is striking is if we now go to a cancer cell, let's say triple-negative breast cancer, one of the major killers of women, what you now find is that those condensates that are controlling genes in that cancer are wildly huge. They're much bigger, encompass much more DNA than we see in normal cells. And this and many other discoveries about how cancer cells are dysregulated have led us to think differently about how we might develop new therapeutics that might have benefit for patients.
Previously, when people looked for inhibitors or drugs, they would look at individual proteins. You do a specific drug and you'd find out how to interact with a specific protein. But now we're saying you need to think about where these drugs interact with condensates.
If I understand it, then I can reproduce it and I can manipulate it. And I can ultimately develop drugs or other approaches to use that revolution or new idea to actually change a system or improve the lives of a patient, translate it into applications in society. That's where biotech stands, between the discovery mode of a new concept or a new technology and the translation of that discovery into something that benefits patients.